company is called Neurix. They work in stem technology and in vitro neural tissue engineering, and the presentation is by Mathurin Baquié.
Thank you, Bruno. Ladies and gentlemen, good morning. My name is Mathurin Baquié. I'm the co-founder and director at Neurix. It's a Geneva-based company specialized in disorders of the nervous system. And as you have seen this through these 3 days, it's a very complex topic. And here's an illustration from the Boston Children's Hospital, which is considered the best children's hospitals in the States. And here's the illustration of the complexity in the nervous system. While the need for therapeutics that target the nervous system is growing, recent results in diseases like Alzheimer's and autism have disappointed. And why such disappointment? And they have one answer. Here it comes. Well, the frustration is that drug candidates that have shown promise in animal models have not demonstrated efficacy in humans. So if we summarize what has been said, most of the preclinical tests performed on animal models lead to failures at the level of clinical trials, meaning not many novel drugs on the market and reduced return on investment. So here at Neurix, we provide a solution. The solution is an alternative to animal testing. And we believe that if you have a human application, you need a human test. And how can you get this human test? Well, thanks to pluripotent stem cells. And to accelerate the process, we generate neural models from these pluripotent stem cells. Here's the principle. You have different patients suffering from different disease. From each patient, you can generate pluripotent stem cells. From these stem cells, you can amplify to a large extent, freeze them, and make different aliquots. Then, when required, you just throw an aliquot and stop the proliferation. Then you say, differentiate into neurons, but also supporting glial cells. So dopaminergic neurons, glutamatergic neurons, etc. But also to understand the aspect of, oh, you have astrocytes. What's the impact of astrocytes on neurons? What's the impact of microglia on neurons? But having a model is not enough. You need to define readouts. You need to define assays. And this is where we are. Not just one assay, but a combination of assays. And we call it a test battery. And through this test battery, you generate a high amount of data, meaning that you have to analyze it through bioinformatics. And here's the added value. The added value is to provide information about neuroactivity, neurotoxicity. If you have a drug, you know the structure, the chemical structure of the molecule. Then at Neurix, we can provide the neuroactivity of the molecule. You are a chemical company this time, and you have a pesticide. You wonder, what's the neurotoxicity? And we can provide this information related to the chemical structure. So this is really to try to combine different information together to, in the end, predict the toxicity, predict the activity of the molecules. So Neurix is a service company. The principle is to address neuroactivity or neurotoxicity questions in a two-step manner. First step is automated screening. So a large amount of molecules that you screen in an automated manner, and you have neurons and glial cells together, and you can get quantitative readouts to end up with the most interesting molecules. Then this is the second step, to have the refined testing, to understand which pathway is activated, what is the most predominant target which is useful for your drug. And in this approach, we generate mini-brains. The mini-brains are neural tissue, so made in 3 dimensions, interconnected neurons and glial cells. But as I said, those cells also from different patients, meaning we can generate different disease. And so far, we have generated a cancer model, which is called the glioblastoma, and also different models of infections, neurotropic infections. And also now we built a model of Alzheimer's. So the principle and the long-term perspective is to have a large panel of patients. From these patients, making a large panel of mini brains. And with those, you can end up with the clinical trials in a dish. So anticipating potential failures in a regular clinical trial in a dish. So this is a way to accelerate drug development. So far we have nice sponsors from academia, but also from private entities all around the world. And as we are based in Geneva, of course, we have a strong collaboration with the hospitals, university, and the engineering school in Geneva. And of course, having sponsors is nice, but also is having a team and having different flavors in the team. So people with finance backgrounds, people with more, like physicists, and at the same time, physicians, and also electrophysiologists, all combined together to make it successful. So this being said, I would like to thank you for your attention and ready to answer your questions.
Thank you, Mathurin Baquié. Thank you. Please, members of the jury.
Can I start? You emphasized that technologically the differentiator was bioinformatics. Have you already started building some knowledge base?
So far, the principle is to work at different levels. So to combine the models. So we work a lot on defining the right models, because if you take the example of infections, some are relevant at the early stage during differentiation, so neural differentiation, and others at the very late stage. So we have first to build the right models. And then at the same time, we screen also a lot of molecules now to build the database. So it's really two approaches, and so far we are in the process of going into having a large amount of data and how to automatize this also.
Could you maybe give an overview of the type of toxicities you can assess?
Okay. So actually, the big question is, how do you define neurotoxicity?
Exactly. Yeah.
And actually, most of the time when you realize what is already on the market, it's more about cell death. And most of the time, you don't want to understand, well, I want to treat with the drug. I don't want to destroy the neurons. I want to define exactly what's the impaired functions of the neurons before cell death. And this is exactly where we are, defining of panels, different assays, in order to define the early impairment of the neurons, and early, much early than the toxicity. So when you define a range of a drug, so this range is the similar in the brain of the patient, and we are able to match this, meaning that in the patient you have this concentration, you know that it's active or neurotoxic. And in our model, we have an example with sitagliptin, which is known to be very safe. It's an anti-diabetic drug. It's known to be safe in the brain. And this is exactly the same we observe in our models. We have like a flat curve, meaning no toxicity. And if we take an example like valproic acid, which is known to modulate mood disorders, in the concentration range, which is similar in the human brain, we have exactly the same. It starts to impair the functions of the neurons, but it's not neurotoxic at this stage. So we really try to combine information from animal models, information from patients, and try to make the bridge shorter. So from preclinical trials on animal models, human beings, and with in vitro models, we try to bridge this in a safer manner.
Mm-hmm. More questions? Yes.
What's the feedback of your customers of BASF, Glaxo, Sanofi?
So the principle is to have those sponsors, because at the beginning we had our own approach, defining what are really the demand, the needs. But thanks to calls, you are able to find out what the industry really needs. And this is the very collaborative approach. They define some endpoints that they want us to deliver, and we are in this process. It's really a collaborative approach. They have a demand. Some points we can address, others maybe not. But it's really to try to improve. They already know that with the animal models, the throughput is limited, the results are limited. So whatever we can provide is always better to what you can provide with the animal models.
Okay. Sure.
What is the single most important benefit you bring to your customers?
So regarding the customers, the principle, as we mentioned, is accelerate the process. If you think about the long term it takes to find out the drug, most of the time it's a failure. And our approach is to de-risk, to consider, to raise the flag before the regular clinical trials and say, we have a human model, we observe that in this in vitro model, there is a potential neurotoxicity. So maybe it's not wise to go into a clinical trial.
Just to be more precise, compared to the model today, which is the rat, how much you can accelerate this step?
So the approach is to combine. The rat, for example, is an organism altogether. So our approach is to be complement. The rat, you see the organism as a whole, you see behavior. With our approach is to define within the cell which pathway is impacted. Within the cells, which is the interaction between the cells impaired. And with the animal models, you cannot address this in a very speedy manner. With our approach, at the level of a mechanism, you can define which target is most relevant, which mechanism is impaired. And so far, there is no really an option today. So we are here to solve and add something which is, we believe, highly useful.
Mathurin Baquié, merci beaucoup.
Thank you.
You.